All right. Hello, welcome. Thank you for joining us today for today's special webinar session called Vaping, Edibles, and the Developing Brain. What can we learn from animal models? My name is Chantel Lincoln, and I serve as a Deputy Director of Drug Free America Foundation. This webinar is supported by a grant we have through the Florida Department of Health and is part of our Marijuana, Pregnancy, and Other Vulnerable Populations educational project. As part of this initiative, we have a variety of resources to support your learning, including this educational webinar series, a free virtual course related to marijuana, and a variety of resources that are available for digital. And if you reside in Florida, we can actually print and ship them to you at no cost. We also have a specialized marijuana track coming up at our National Prevention Summit in February of 2026 in downtown St. Pete. So include all this information along with a certificate of attendance and today's recording in the follow-up email. A few housekeeping items to note. This is a webinar, so everybody is muted except for the presenter. Please, in the chat box, which I see some of you already have, please introduce yourself, put your name, organization, and where you're from, as we love to know who's joining us today. If you have any questions throughout, please place them in the Q&A box, and that way we'll address them with the presenter. Now it's my pleasure to introduce our speaker, Dr. Khokhar. Dr. Khokhar has completed his undergraduate training at Queen's University and his PhD in pharmacology and toxicology at the University of Toronto. He then completed a postdoctoral fellowship at Dartmouth College and has held prestigious awards, including a CIHR postdoctoral fellowship and the NIH Pathway to Independence Award. He is now an associate professor and Canada Research Chair in Translational Neuropsychopharmacology at Western University. His lab has published over 65 papers and received nearly $6 million in funding. Dr. Khokhar's research focuses on vulnerabilities to substance use, from genetic risk to developmental effects, and he has developed innovative methods to study emerging routes of drug exposure, including vaping and edibles. Please join me in welcoming Dr. Khokhar.
Thank you so much, Chantal, for the introduction. Great to be here and look forward to chatting with everyone today. And so today we'll be talking about vaping edibles and the developing brain and what can we learn from animal models. And before I start, I would like to acknowledge that the work I'm about to present was done on the traditional lands of the Mississaugas of the Credit First Nation, of the Anishinaabe people, the Haudenosaunee, the Lenape and the Chinooka nations on lands connected with the Dish with One Spoon Covenant Wampum. And for those who might not have heard traditional or indigenous land acknowledgements before, it's an important reminder for us, especially me as a neuroscientist. I try to figure out these most complex mysteries of our brains, but we often forget that neuroscience is done by flawed human beings, us, and has been and continues to be used to uphold systems of racism and colonialism. And this is not something we shy away from, but instead see how we can contribute to both mitigating, understanding and mitigating the long-term consequences of settler colonialism and genocide on the brain, body, and society here in Canada and across the globe. And, when it comes to cannabis, that again is an important remembrance to have. Cannabis in Canada is a big thing. Even before we legalized cannabis in 2018, we were some of the heaviest users of cannabis worldwide. And that's kind of why we haven't really seen much of a change in the amount of cannabis use in Canada. So this is past 12 months use. And so you can see, in Canada, this is most of the listeners today are probably in the US. Canada provides like a look in the magical ball or as a prediction of the future as things move towards legalization or rescheduling, at least maybe in the US. And so in the past 12 months, more than a quarter of Canadians used or reported using cannabis. When you look at the specific age demographics between 16 to 19-year-old, that's just over a third. At that 20 to 24 year mark, half of Canadians reported using cannabis in the last year. And then as that age group increases to above 25, around a quarter of Canadians use cannabis. And what's interesting is cannabis, when compared to other commonly used legalized substances, cannabis seems to have a better PR. more of a less of a perceived substance use risk, right? Like there's tobacco smoke right there, 95% doesn't really get worse than that. Single largest preventable cause of death worldwide. We know it's bad. But then e-cigarettes, which have really picked up, and vaping has become a thing largely because of its safety profile, right? Like it's safer than tobacco smoke, not the highest bar, but still safer than tobacco smoke, but it's still coming up at 87%. And then we look at alcohol. Alcohol remains a big problem. The only safe amount of consumption of alcohol is no alcohol at all. And yet we still see it, show up at around 77%. And then cannabis, especially in forms like edibles, only 64% of Canadians thought that there was any risk associated with it. And so it's really important that we try to see where public perception, where it might not align or where it might be misaligned with the actual evidence. And so this is something we've been trying to do in the lab, is figure out those causal things. And cannabis, because mm, it is a thing that um, can be very polarizing. It's important to then lead with evidence and really try to establish causality. What does cannabis cause in the brain versus what is just an association? And so today, what we'll do is think about, because of how polarized it is and how anything to do with cannabis gets picked up by the media and talked about in a variety of ways. What we're going to do is take some recent headlines or vignettes or social media posts that have happened around cannabis and try to get to the science behind it and try to really understand what it is that is real behind it. And so we'll start off with cannabis and the developing brain. Um, you know, we've talked about cannabis and the developing brain for a long time, all the way back to this is your brain on drugs. Fried eggs don't really have much to do with cannabis, but maybe some people want fried eggs after cannabis. But that approach of trying to essentially scare kids out of using cannabis hasn't changed. Even as more recently, this is the Stoner Sloth campaign out of Australia. For those of you who haven't seen it, kind of like the scene from Zootopia, where at the DMV, right? And so that it suggests that cannabis makes you like a sloth, and when you look at the evidence, it's actually not the case at all. While there's a slowing down of motor behavior, there's actually increased impulsivity. And so paradoxically, you actually see two opposite things happening, and there might not be an actual change in how somebody reacts. But that hasn't stopped, these mass media campaigns to try to use this to stop people from using cannabis. So, as I was mentioning, going and looking at the trends that have appeared in the last few years, it's important that we try to understand what they mean and what that means for the developing brain. And so, as we've seen recently, teenagers who use cannabis are shifting from smoking to vaping. So, what does vaping do? How is it different from smoke cannabis? And how do we understand its effects on the developing brain? We know from a lot of published literature in humans where they use drugs during adolescence. So here, if you were to look at the regular trajectory of the developing brain, adolescence is this unique period in which any exposure to any drug, so it could be nicotine, cannabis, opioids, or alcohol, changes the trajectory of the brain development and increases the risk in three domains. The first one is substance use disorder. So developing a substance use disorder either for the same substance. So if you use nicotine, then going on to become a combustible tobacco user. Or if you used cannabis, then having a cannabis use disorder later in life. That risk increases. In fact, the earlier you start, the greater the risk. The later you start, the lower the risk. But There's also cross sensitization, where now use of one drug may increase the risk of developing the substance use disorder of another drug. The other association that we see is psychopathology, where now individuals who use cannabis during adolescence may be at a greater risk for developing schizophrenia or anxiety or depression. And then the last one is cognitive deficits. And again, we see that across most of the drug classes, this use during adolescence does produce these long-term changes in cognitive function as well. The problem with these studies is either they're cross-sectional, or even now that they've done, we have the ABCD, the Adolescent Brain and Cognitive Development Study happening in the US right now, where 10,000 12-year-olds were recruited, and they're being scanned every 2 years, and all of these outcomes are being looked at. Even then, those studies remain associational. It's possible that the group that uses drugs was always different from the group that doesn't. And so we need to be able to establish what are the causal effects of these drugs, especially cannabis, on the developing brain. And that's where animal models come in. It's not really ethical, and you can understand that, to give one group of teenagers cannabis and another group of teenagers and no cannabis, and then see how they turn out. But importantly, in addition to that, we can't really get a look under the hood, right? The skull remains the protective covering for our brains, and so we can't really assess what's going on in the brain. And that's where animal models come in, and they help us understand. And we know a lot about the brains of animals. And then the really nice thing is a lot of these systems that drugs act on in the brain are actually highly evolutionarily conserved. And so you have the same receptors expressed in the brains of monkeys and rats and mice as you do in the humans. And so, and as a result, the effects that you see in humans, you can actually recapitulate them in animals, and then also use animals to predict those long-term effects. And this is a really busy slide on purpose. What it's trying to show you is that adolescence, regardless of which system you're looking at, the dopamine, the glutamate, the GABA, the nicotine, the cannabinoid, the opioid, they're all going through a lot of flux during that adolescence period, making this a uniquely vulnerable period. It's sort of, think of it as a prism, right? And where now this is a period of change, and any exogenous influence, anything from the outside, could be nicotine, could be alcohol, it could be cannabis, now is going to shine or get refracted through this prism, where now the effects on the long lasting effects are going to be viewed through this period of change. And so what we've done is try to really, for a long time, people have been injecting rats and mice and monkeys with drugs. That's not how people use cannabis. And that's one of the nice things about being in Canada with legalized cannabis. I can literally just go to the corner store, buy the cannabis store at the corner of the street. There, it's not available at corner stores, thankfully. But buy the cannabis. I actually go and ask them, what's your most popular cannabis? And then we buy that, and then we expose the animals to it. And here we use a volcano vaporizer. It's the OG vaporizer. It's an oldie but a goodie. In that we can take dried cannabis flower, and then vaporize it and expose animals to it. And here we use cannabis that contains high levels of THC, a balanced THC and CBD, and then a predominantly CBD strain as well. And CBD has had, again, very good PR in that it's only good and has no negative effects. And so it has medicinal properties. And so it's really important to then put that question to the test as well. And then what we do is expose animals. So the PND 28 to 42, that's the period of adolescence. Then this is the really nice thing about animal studies. That's 14 days. That's the entire adolescence period in a rat. Then you give it another 20 days, and now the animal is a full adult. So within the matter of a month, you've basically covered the entire developmental trajectory. And now you can look at the effects on the brain, you can look at the effects on the behavior, and see what is changing as a result of cannabis alone. Now you're not having to worry about any socioeconomic or socio-demographic differences. You're not having to worry about early life trauma. You're not having to worry about all of those factors that would confound these studies in humans. You can purely look at the impact of cannabis. And so, what we did here is we took the animals and we exposed them for these 14 days, once a day, every day for a short period of time. And again, what we're trying to do is really not only mimic the route of administration, so here vaping instead of injecting animals, but also the amounts that are generally used by adolescents. Like if you inject a rat with a honking big dose of THC, is that really representative of what a human adolescent might use, somebody who's just starting up? And so we really, once a day for these 14 days and very small amounts. And so here you can see the plasma levels. So reaching, 10 to 15 nanograms per mil, which is basically equivalent to a puff or two of cannabis on these days. So we see that the high THC cannabis produces the highest levels of THC. The high CBD cannabis produces the highest levels of CBD, and the high THC cannabis also produces the highest levels of 11 hydroxy THC, which is the active metabolite of THC. We also see that the drugs are biologically active. They result in decreases in locomotion, or how much the animal moves, changes in body temperature, as well as changes in tail flick latency. So which is the measure of how much pain an animal perceives. And so cannabis reduces that. And so we see that with the high THC cannabis. Now, when the animals get into adulthood, now when they're older, when they're, now they've gotten to a point where they're full adults, we assess learning in these animals. And there are two types of learning that we're assessing. One is like an appetitive learning. It's called a PCA task, or a Pavlovian conditioned approach task. What it is basically seeing how well an animal learns to associate a lever with the food. It's kind of like when we drive by McDonald's. The golden arches have never given us food, but over time we formed this association that golden arches means Big Mac. Big Mac means I'm hungry, pull into the drive-through. And so this is essentially what the animals are learning as well. Lever comes out, lever goes back in, a sucrose pellet is delivered. Over time, the animals learn that this lever has something to do with the food. So then they start to gnaw at, bite at, chew at that lever every time it comes out. And what we see is that here are the control animals, that they just got like hot air blown at them. Sometimes happens to us too. Um, and then we see that they learn this just fine. They're learning this task beautifully. The balanced THC and CBD actually also learn this beautifully. So, there's some sort of effect of THC and CBD happening where they might be canceling each other out. But when you look at the high THC and the high CBD, both of those groups never learn this task, suggesting that there might be a learning impairment that's arising directly from that exposure to cannabis during adolescence. Then we looked at another task. This is an aversive learning task. Here, an animal learns that if a light appears or a sound is given, they are going to receive a mild foot shock in the seconds that come after it. But if they can run across to the other side, they will not get shocked. And so as we can see here, the control animal learn this very quickly. This is within one day, within 1 hour, they're effectively able to avoid all of the incidents of the shock, not the animals exposed to cannabis. All of them, even the balance group that was protected in the appetitive learning task, still can't learn this one. And so it's really important that we assess a variety of types of learning, but also doesn't matter if it's THC or CBD or balanced THC and CBD, all of them disrupt learning. What we also looked at is a pre-pulse inhibition task. This is often used as a way of modeling schizophrenia or psychosis-like behavior in animals. And a lot of people in the past who've said, "Oh, cannabis causes schizophrenia," especially from animal studies, have actually used this as a measure. But then again, as I was mentioning earlier, they use honking big doses of THC. Here at these moderate doses, we actually see that this assay, which essentially looks at how well you can modulate or gate your response to a startling stimulus. So if you walked in through a door and somebody jumped out from behind the door and surprised you and said boo, you would startle. But in the process of doing so, if they kicked the door or knocked over a garbage can, then that boo would be less startling for you. And so we can do the same thing in animals. They get like a loud stimulus. Before that, they get a smaller stimulus. And normal animals will reduce how much they get startled by it. And here we see that cannabis exposed animals do just fine on it. Uh, it's important to look at behavior, but it's also important to look under the hood and see what's happening in the brain. And so for that, just like we use animal exposures that are more akin to what's done in humans, our readouts of the brain are also in line with what we use in humans. And here we use an MRI. Yes, it is an MRI for rats. That's where the MRI goes in. So it's tiny, and there's like a little bed that they get placed on, and then they get inserted, unlike the bigger MRIs that are used for humans. And what we see is that it is, in fact, the CBD exposed animals that have the most marked changes in how well the connections in these brains exist in these animals. That the CBD group had higher functional connectivity compared to the control group and compared to the THC group in parts of the brain involved in somatosensory, so sensation and visual and auditory sensations in the body. We also see that not only is the functional connectivity changed, but also the structural, the highways that connect these parts of the brains are also changed, where we then see more connections between the parts of the brain that are involved in addiction and fewer connections that are involved in locomotion and functioning, cognitive function as well. What's interesting is the data that I was presenting so far was done in male rats. When we look at female rats, we actually see that they're protected against those effects of on learning across all three types of cannabis. So it'd be important to figure out why is it that females don't have the effects the same way that males do. And we have a couple of things that we think might be happening, and so I'll show them here. The first one is that the amount of cannabis or the THC and CBD in females ends up looking very different from the amount in males, even though we use the exact same cannabis down to the batch number, which suggests that the female body receives and perceives cannabis differently from the male. And so it's important that we try to figure out why is that and study that some more. In addition to that, we also see that those behavioral effects are very different. Only the high dose of cannabis produced that change in pain perception, whereas all three types of cannabis increased it in the female. So there's differential sensitivity as well. One thing that we think that might be contributing to this is that there are these cells in the brain. These are the resident immune cells of the brain. They're called microglia. And we think that it's the sex differences in how these immune cells function within the brain that might be contributing to these differential effects. In males, we see that cannabis reduces the number of these microglia in the brain, but not in females. And so it's possible that there is now more immune activation, or now the males are more susceptible to infections or other things as a result of the cannabis, and that might be contributing to those long-lasting changes in learning. And then the next thing that going through the different news stories that have come up. The next news story that I wanted to talk about is withdrawal. Withdrawal for a long time has been considered like cannabis withdrawal has been enigmatic in that some people don't believe it's real. Others don't think it's as severe as what we see with other drugs. But if Snoop Dogg can report having symptoms of cannabis withdrawal, then it's probably something that we should study and try to understand why is it that um, cannabis withdrawal and how do we mitigate it? How can we come up with a drug that can reduce the symptoms of withdrawal? And so we've again modeled this in animals. So if we expose animals for 7 days, 3 times a day to the same cannabis, high THC cannabis that we gave earlier, we see that these animals show increases in withdrawal across a variety of symptoms. So um, total withdrawal scores, but they show head and body shakes and tremors and blinking, etc. And we also see that we can both see it in animals that are not exposed to a cannabinoid receptor antagonist, as well as animals that are just exposed to saline. In that, just the passage of time, when you take away the cannabis, withdrawal shows up. Moving on to another uh, news report that we often see, police warn of cannabis edibles that look like candy ahead of Halloween. When I was younger, it was razor blades, and so now it's rainbow fentanyl and cannabis edibles. But that doesn't mean that the risk isn't real. In fact, in Canada, they actually almost did an experiment of sorts where one of the provinces did not legalize all types of cannabis. The only type of cannabis they legalized was edible that would not be appealing to kids at all. So none of those, Sour Patch and High Chew or whatever. You could only have like rutabaga flavored edibles or dates infused with cannabis oil. And what you see is in that province compared to all the other provinces that legalized candy like edibles, there was twice the amount of cannabis poisoning in the other provinces compared to Quebec. And so good policy matters. And that's where I think all of everybody here in attendance, you are the sort of warriors that can fight on that front, and that good policy can make a big difference. And in the US, I didn't even realize that percents could get this big, but in the last 5 years, the number of kids who have accidentally ate cannabis edibles has jumped 1,375%. In Canada, even the teen use of cannabis has increased edibles by 43% after legalization. So making this an important topic to study. And so again, just like with the vaping, we wanted to keep the mode of administration as similar to humans as possible. And that's where the amnesia comes in. They have a recipe for cannabis into infused Nutella, and we copied that exactly using this Five Founders cannabis oil that's also available from the cannabis store, and let the animals eat the Nutella, a pretty high dose of Nutella to really model what um, the effects would be like if you ate a chocolate bar, all of it, without knowing. And oftentimes that's the case, right? Because it doesn't hit you immediately, you might not know for a while that you've actually taken too much of an edible. And so we looked at behavioral effects, we looked at brain communication by collecting electrical signal from the brain, as well as measuring the levels of cannabis in the brain and in the plasma. And we see again a big sex difference here, but we see that high levels of THC are produced after this, compared to the vaporized, the edible produces higher levels of THC and 11 hydroxy THC, which was around 3 to 4 nanograms per mil. Now it's over 40 to 50 nanograms per mil, especially in the females. So a much higher dose is achieved. But in addition to that, you have much higher concentrations of THC in the brain and 11 hydroxy THC in the brain. We also see long-lasting behavioral effects. Body temperature stays down for 24 hours, increase in that pain tolerance for 24 hours, and the amount the animals move is also reduced 24 hours, up to 24 hours later. We also see that the activity in the brain is significantly affected. Even up to 24 hours later, there are significant reductions in gamma power, which is one measure of these longer ranging communication signals in the brain that are significantly impaired after cannabis exposure. And so we've now taken this, and as the New York Times recently reported as well, cannabis poisonings are rising, mostly amongst kids. The other two populations that actually are affected are older adults and pets. And so, studying poisoning is important, and trying to see if we can come up with treatments for it is also paramount. So here we took juvenile animals, so postnatal day 35, exposed them to cannabis, and unlike the adult animals that I was showing earlier, these effects last even longer. One single exposure to an overdose of cannabis edible results in effects last up to 14 days when it comes to, how much the animals are moving, or even that pain perception signal. And even when these animals grow up and they're exposed to cannabis again, you actually see an enhancement of the effects of cannabis. So here, these are animals that were previously exposed. These animals were not previously exposed. A re-exposure to cannabis actually now changes how they respond, both in terms of the distance moved as well as tail flick latency. So now getting to the last part of this talk. What is the role of genetics? While we now talked about, the role of development, are some people just more likely to use cannabis, or are they more likely to achieve or have adverse events as a result of the cannabis? And that's where genetics comes in. This is a recent Instagram post where they suggested there's this gene. In fact, one of these genes associated with cannabis use, called cadm2, has been linked to is what makes weed addictive for people. And so we decided to try to answer that question. Well, if it's on Instagram, it must be true, right? So it's important again to assess what the truth is. And yeah, obviously, they're basing this on real science. And one of these papers is the Pasman 2018 paper, where they found that cadm2, this is what's called a Manhattan plot. These are all of the genes that are significantly associated with cannabis use. This is lifetime cannabis use. So, have you ever used cannabis in your life? cadm2 shows up as a pretty significant hit when it comes to that. So, since half of genetic of variance in cannabis use is explained by genetics, it's important we try to figure out what is the role of this cadm2 gene in cannabis use. So since then, we've actually looked at how many days did you use cannabis during your heaviest 30 days? Again, looking at a frequency of cannabis use, and we see again that cadm2 shows up as a significant hit. In fact, the only significant hit that shows up there. What is cadm2? It is a cell adhesion molecule that is associated with drug use, cognition, psychopathology, metabolism, and neurotransmission. And just like I was saying with the neuroimaging studies and the association studies, we don't know for sure if cadm2 actually plays a role, or if it's just an association that has come through another mediating pathway. Maybe this is associated with race or socioeconomic status or impulsivity or something else. And that's what then shows up as a signal in the cannabis use genome-wide association studies. And so what we did here is took these animals and actually created animals where we knocked out this gene. So now these animals don't have the cadm2 gene anymore. And so now we can see, does that change how they respond to cannabis? In humans, it's the higher expression of cadm2 that makes somebody more likely to use cannabis. And so if we remove it, does that now make this animal less likely to use cannabis? And again, we use cannabis that's available from the Ontario cannabis store. So we use cannabis oil. We made cookie dough balls. And so we put two balls, cookie dough balls, in their cage, and the animals tell us which one they prefer or how much by telling. They vote with their feet and their teeth, I guess. Where if they eat more of the cannabis containing one, then we know they prefer it. If they eat the regular cookie dough, then we know that they prefer the one without the cannabis. And this is where we find that knocking this gene out significantly reduces how much of the cannabis containing dough these animals eat. In this case, it was pure THC. In this case, it's cannabis oil. This has CBD, terpenes, full spectrum, but has the same amount of THC as the pure THC we saw earlier. And again, we see knockouts don't like it. Animals with the gene like it, and then animals that have half as much of the cadm2 as the dose of THC increases, they do not like it at all. What's really cool here is with the cannabis oil, females prefer the cannabis containing dose significantly more than the males. And what's really cool there is while most types of cannabis in humans are male dominant, when it comes to edibles, those are used in a greater proportion by females compared to males. And so our mice are showing the same effect that you would see in humans. So what we can do is assess, why is it that they're responding so differently? Is it hitting them differently? Or is the body modifying or seeing it differently? Does the cadm2 knockout have more of it or less of it? And that why the differences are occurring. And so the first thing we did was assess the behavioral effects of the drug. And what we see that in THC, we tried a low dose of THC and a high dose of THC. We see that low dose of THC doesn't really do much, but a high dose of THC really reduces body temperature in the wild type and heterozygous animals, but the knockout animals don't show that. So they're almost less sensitive to the effects of THC. On the other hand, when we look at that anti-nociception, how much pain do they perceive? We see that the animals that were knockouts actually show an enhanced response to the THC compared to the wild type and the heterozygous. And then last but not least, usually when you give cannabis to animals, you see that they move around less. But not the knockouts. When they're given cannabis, even at a high dose, they actually move around more. So it's almost as if the drug is now having the opposite effect in these animals. Another measure we looked at was pre-pulse inhibition. That's what I was mentioning earlier, the boo and the sound before it. We see that knockout animals already showed disruptions in that pre-pulse inhibition, and that when we add THC to the mix, even those half deleted gene animals now show disruptions in their pre-pulse inhibition. Lastly, when we looked at the pharmacokinetics of it, so how much of the drug gets into the body of these animals, we actually see that there are no differences between the three different types of genotypes of cadm2. So it's not changing how much of the drug gets in the body or gets out. But what we are seeing is that males and females, yet again, this is the third study that we're seeing this in, both receive into the body cannabis differently and process it differently. And, maybe anecdotally, we've known this all along, but it's really important that we see what those differences are. Why are women more susceptible to the effects of cannabis compared to men? Why is it that we are seeing differences in how those long-term effects after adolescence also happen? And so it's important that we try to establish where and how these sex differences arise. The other thing that we've done with these animals is try to figure out what is happening in the brain. And so this is again using MRI in the brains of these animals. And we see that the knockout animals compared to the wild types have reductions in, or sorry, increases in um, connectivity of the midbrain, a part of the brain that previously in humans with the cannabis use disorder is thought to be over connected or overactive. And so what we see is that these animals that weren't preferring cannabis show the opposite signature of those humans in that they have lower connectivity of the midbrain. The humans that higher connectivity of the midbrain if they preferred cannabis. These animals don't prefer cannabis and they show lower connectivity of the mid. So I hope thus far I've shown you how the different ways we can study vulnerabilities to cannabis in animals and using animal models, but also how we can begin to get at what those causal effects of cannabis on the brain, on the developing brain, on the brains of males versus females are, and how we can study these effects using animal models. I wanted to end with some future directions. We now have rats that can self-administer cannabis. So instead of just, hot boxing them with the vaporizer, the volcano vaporizer, what we can do is let them decide how much cannabis they want. They can go and press a lever or go poke their nose, and then they'll receive a plume of cannabis. And they can decide, I'm done now, I've had enough, or they can continue going, and then we can assess the long-term effects of this on the developing brain. We've also now started doing some studies using marmosets. The cool thing about marmosets is they have two sets of twins every year, and so you can really control for genetics, control for rearing environment, and look at the effects of the drugs on these animals. In addition, we're also testing the effects of blocking the cannabinoid receptor in the brain to see if that can be used to undo the effects of cannabis poisoning on the brain, as well as trying to see if we can get cannabis to its inactive carboxy metabolite, if we can get the body to kick out cannabis more easily, does that reduce how long that poisoning effect lasts? And with that, I'd like to thank the people in the lab who do all of the work and our funding sources, and to all of you for being an attentive audience. And I'd love to answer any questions that you might have.
Well, thank you so much, Dr. Khokhar. This was really fascinating presentation and so important. And I would love to have you back on to hear more once that future research is done. But we've got a lot of comments and questions. So if you have a question, we're going to move right into it so we can address as many as possible. Please place them in the Q&A box. We have some that came in the chat, so I'm just going to start from there. The first one is, have you used higher percentage of THC above 20%?
Could you say that again? Sorry.
Yes. Have you used higher percentage of THC above 20%?
Yeah, so we have, for example, right now, one of the students was actually just messaging me this morning about a strain called Pink Kush, and it's at 35%, and so we're going to test that out in the lab soon. But in addition to that, we've actually been testing shatter and dabs as well, and so those are 90% and higher, and looking at those effects, and so we do have some data on it, and it seems to track. The more THC you have, the longer lasting the effects and the more of a hammer that you're hitting the brain with.
That makes sense. Thank you. The next question is, do you check for contaminants in the products you purchase and use?
Yeah, no, that we haven't done. The only thing we have done is confirmed the labels. So when it says this is a high THC, we will then go and confirm it. But we have not looked at the contaminants. But the benefit of being in a legal market is that now is the government's and the uh the producer's responsibility. right? And so they're like the illegal market comes with all sorts of with the poison drug supply problem that we're dealing with, right? But that's one benefit of the legal cannabis is that those contaminants hopefully are being avoided.
And then are the mice from the same strain genetically similar?
They're supposed to be. So, and that's a great question. We actually have, so all of the studies I presented today are inbred strains. So these are supposed to be as genetically identical as possible. But we have now started working with a heterogeneous stock rat, where they took essentially 9 different strains of rats and crossbred them to really create like a diversity of genetics in those rats, and we're testing the effects of cannabis in those rats as well. So hopefully the next time around I'll have some more of that data.
Wonderful. Next question. Do you know if the knockout rats exposed to THC as teens differ than the non-knockout rats on learning in adulthood?
Yeah, no, that's a really good question. And one of the students in the lab is actually doing that just now. Is because what I presented so far is just acute effects of cannabis. Do they find it rewarding or not? But are they differentially susceptible to the effects on learning? We don't know, but hopefully soon we will.
Wonderful. Let me see. When children accidentally ate the edibles, how much is the effects of damage in the early developing brains?
Yeah, that's exactly what we're trying to figure out. So I showed earlier that if they're exposed in adolescence and then we test them again in adulthood, are they perceive cannabis differently? Like it hits them differently. But now we're also trying to see if that affects learning. And like this has been the hard part. Um, every time I've tried to get funding to do some of this, people are like, one exposure to cannabis couldn't do so much harm, right? Like, oh, what's one exposure to cannabis going to do? And I keep trying to say, like, actually, it's a hammer, and so I haven't had the best luck getting that project funded, but we're still gonna find a way to do it. So hopefully, I'll have some of that data.
Thank you. The next question, do you have data on marijuana use and violence? One of our attendees put a link to a study, and it says this study shows difference between males and females.
That's really cool. So violence is a little bit harder to study in rodents, right? Like, ja- but we can assess aggression. And so one of the students, a PhD student in the lab, he came in with real interesting project that he wanted to work on was seeing how cannabis alters social interaction, both positive ones. So between a male and a female mouse, for example, and negative ones where now you have like a bully mouse and a victim mouse, right? And so that's what he's studying right now. He just started in the lab, and so that's the goal is to see how are positive social interactions versus negative social interactions perceived with or without cannabis, and how are they remembered? Like, are you now more likely to forget a bad memory if you had cannabis at the time that you were experiencing it? Or are you now like, is a positive memory going to be more positive because you were on cannabis when you had that experience? And so that's what we're hoping to study right now.
The next, pregnant moms who use THC, what is the impact in the new baby's brain? We have DCF referrals of moms and babies positive for THC after birth.
Yeah, so that's a big question. And while we're not studying it in the lab ourselves, there are a few labs. So Matt Hill's lab at the University of Calgary, Ryan McLaughlin's lab at Washington State University, Yasmin Hurd's lab at Mount Sinai in New York. They are studying it. And they've seen a couple of different things. They've seen that definitely impacts body weight. So body weight is much lower in animals that were exposed to THC in utero. But also they're seeing long-term changes in cognition and even autism and like behavior in some of those offspring as well. And so there are effects of adolescent exposure.
There is a comment that said we just
Sorry,
did a
not
study
adolescent. Mhm.
prenatal.
Yeah.
We just did a study in New Jersey. An attendee said that is a regulated state. It showed that 41% of regulated products were mislabeled regarding THC content. Let me see. Your data is very valuable. Please publish it. We have a comment. Are these impacts the same as those of studies with alcohol and adolescent mice?
Yeah, so that's where our lab gets where we're kind of drug agnostic. So we do alcohol, nicotine, cannabis, psilocybin. We test it all on the adolescent brain. And so there are differences and similarities. Alcohol seems to act differently on the brain than cannabis. Cannabis changes how impulsive an animal is going to be or how interested it is in learning a task, whereas alcohol seems to make the animals just be more prone to making errors. Nicotine makes animals more likely to sign track. So that graph I showed, nicotine actually changes it the other way. But I think depending on which drug it is, the effects, your mileage may vary. But the other important thing that we try to study in the lab is when drugs are combined, right? Like polysubstance use is the rule and not the exception. You'd hardly find a person who says, I only use one drug and I don't use anything else. And so we've actually looked at alcohol and cannabis, nicotine and cannabis, or nicotine and alcohol, and seeing what the combined effects of the drugs are as well.
And what percentage of THC should consumers be warned about psychosis? There's a pending lawsuit in progress on psychosis.
Yeah, no, I think there's two factors, right? Like, so while there's what it says on the label and what you buy from the store or the dispensary, there's also how you use it, right? And so if you're going to now vape it and keep using it all day and all night, that's going to be a very different dose than if you just tried one puff, right? So I think it tracks, right? Like it's dose dependent. And you can get to a higher dose by either increasing the incidence of use or the product that's being used. And so both are important to consider. And so what I always say is start low and go slow and delay for as long as possible. If you can start when the brain's more developed, the less the chance. If you start earlier, the greater the chance. If you start with higher THC forms, the greater the chance. If you escalate really quickly and go from 10% to 20% to 30% to shatters and dabs, then that's another. And then if you have any family risk of psychosis or other serious mental illness, then really like that's when it's a perfect storm.
Absolutely. Are there animal models to measure psychosis that could be used in your lab?
Yeah, so that's I didn't talk about that today, but we one of the things that we study in the lab is the interaction between the chicken or egg question of cannabis use and schizophrenia. So we have animal models of schizophrenia that are created by an early developmental insult, so early life insult in neonatal animals. And we see that cannabis during adolescence significantly worsens that. But we can look at a variety of different outcomes. We can look at how they learn, how they perceive sounds, how they hallucinate or have psychotic-like experiences. Even the pre-pulse inhibition method that I was mentioning earlier is used as a model of schizophrenia because you also see sensory motor gating deficits in patients with schizophrenia.
Mhm. Thank you. I'm going to move over to the Q&A box because there's a few questions over here. It says, I want to better understand why most, if not all, confounding factors can be ruled out in rat studies because they don't experience the same length of childhood, adolescence, and the same traumas. But aren't there studies that do prove rats can be traumatized and learn from certain pain stimuli? Couldn't you examine rats raised in a fairly humane environment versus a simulated traumatizing one and compare how THC may affect their brains differently?
Uh, great, great question and great thought. And you've hit the nail on the head. And that's exactly what we're exploring right now. Not only are we exploring that, so an impoverished versus enriched environment, can that actually maybe mitigate some of the effects? So, for example, like if, well, yes, both animals got cannabis exposure, but one has an enriched environment, has access to a mezzanine floor, has access to a running wheel and toys and whatever, and the other one doesn't even have enough bedding. How do those now affect it? And then one of the students that just started in the lab, she's actually interested, and this actually goes back to my the land acknowledgement that I started with. She's indigenous, and she's really interested in the impact of residential schools and maltreatment and the transgenerational impact of it as well. And so, if somebody went through childhood maltreatment, now, are their children more likely to use cannabis, or are they more likely to then have negative or adverse consequences of cannabis? So, your question fits that line of research perfectly. And so, hopefully, the next time I'm here, I can share some of those findings with you as well.
Thank you. Okay, the next question is, so the link between the CADM2 gene and cannabis use is that it increases addiction to cannabis, increases likelihood of use, or is it just a correlation between that gene and cannabis use?
Yeah, so our data is starting to show that it's not just a correlation. Like those animals now, cannabis hits them differently. So obviously something changes in the brain and the body of these animals that now cannabis no longer has the same effect. Like you would never think that, oh, this person used cannabis and as a result, they're now running around more. It's usually the opposite, right? Think cocaine, amphetamine, yes. Like now they're running around more. But these animals, when you knock out that gene, instead of slowing down, they speed up. And they were already faster to begin with. And so it's like puts them into hyperdrive. And so there's something different. And that might contribute to, and then those two graphs that I showed with the Manhattan plots, the first one was lifetime cannabis use. Do they ever use cannabis? The second was addiction, or how frequently are you using it. So it's more of a measure of addiction, right? And that again, the same gene came in, and so now we're actually using the same mouse model, and we have a rat model now too, where we're letting the animal, they just ate the edible before. We're now letting them press a lever to get a plume of cannabis vapor and see if they differ in that, and to see if they differ in their dependence on it, if they differ with withdrawal. And so hopefully the next time around, I'll have some of that data as well.
So fascinating. I don't see any more questions in the Q&A box. There is one left in the chat box that says cannabis and prescription meds, antidepressants, anti-anxiety, antipsychotics. Any studies showing how these interact?
Yeah, that's where it's also really interesting because you see both um, types of effects. One of the things that I think is important for everybody here to know about as well is CBD really messes with your liver. It inhibits all of the enzymes that metabolize a lot of those drugs. So what happens is if you're taking antidepressants and you're using a CBD oil or CBD containing cannabis, now your body's ability to metabolize those drugs is going to go way down. And so you end up essentially getting a heavier dose, a higher dose of your antidepressant or antipsychotic. And so that's why it's important to tell your doctor if you're using cannabis. Uh, but in addition to that, you actually also see that in the brain, as that was at the liver, but in the brain, cannabis can actually counteract some of the effects or worsen some of the side effects associated with the drugs as well. And so, really important again to discuss your cannabis use with your doctor.
So critical. Thank you. And just a comment that the one of our attendees would like to use you as an expert. I'm going to save the chat, so I'll provide you his email address and whatnot, so you
Okay.
guys connect.
Otherwise, ask them to send me an email and I can respond to them directly. You
Okay.
can find me, I'm easily found with the Google search.
Okay. And is it okay if I share your email address with the attendees afterwards?
Sure.
Email. Okay. All right. Well, I do not see any more questions. You've answered a lot. Your research is so fascinating and it's so important to understand these causal effects. We really appreciate you joining us today and all your magnificent research you've shared and your wealth of knowledge. And we hope that you will come on in the future and share your great research with us.
I'd love to. Thank you so much.
Well, thank you guys for all attending today. I'll send you the certificate of attendance, recording, his information, and some resources I shared in the beginning. Thank you all, and we look forward to seeing you at our next session.
Everyone.